Hello, everyone. My name is Barry Ryan. I'm a PhD student at the University of Edinburgh. So in this talk, I'm going to discuss a bit about how we've used a clinical assessment for Parkinson's disease using LLM to kind of generate embeddings from this and generate patient similarity. Then how does that affect the interpretability? So although this is classified, although we use this as a classification problem, it is really about the enhanced interpretability that we can get from this. So starting off, if there's anyone who isn't really aware of like Parkinson's disease, it pathological hallmark of that, and it's kind of like the end term, I guess, long-term effect of it is just the death of cells in this brain region called the substantia nigra. But I think the common consensus among all clinicians and work with Parkinson's disease is that there are a numerous number of, I guess, molecular malfunctions that then converge on the same set of downstream symptoms. So that's the long-term effect. But in the short term, there's actually this really highly variable set of symptoms that people with Parkinson's disease actually end up going through. And they are the classical ones of tremor, but you also have things called postural instability, rigidity, and then you also have a set of non-motor symptoms. So how can we actually overcome or group patients together with similar symptoms to try and look or identify and interpret the actual hallmarks of the disease? So essentially our methodology here is we want to combine genomic and multiple omic features, but also using a clinical assessment to try and guide which features we want to learn for different groups of patients. So we use the clinical assessments by embedding that in a LLM, and then we generate those, and from those embeddings, we're able to measure our similarity and our distance between our patients. At the same time, we want to perform a multi-omic analysis, and to do that, we use a deep learning framework where we want to just combine different data sets, and then finally we combine those two using a graph neural network, and at the end, we're doing a patient classification problem. But in the medium step, it is what is the interpretability that we can do that. So although it's patient classification, I reiterate, it's the interpretability that we are interested in. So the data which we're working with, there's numerous clinical assessments for Parkinson's disease. The probably most common one and the largest one is what's called the Movement Disorder Society Unified Parkinson's Disease Rating Scale. And this is a clinical assessment with over 50 different assessments or 50 different questions. There's 4 different parts. In this work, we focused on the 2 main parts, that is part 2 and part 3, because they cover the most number of symptoms in terms of the non-motor symptoms and the motor symptoms, and they are kind of in question-answer based pairs. So that's really, really nice and intuitive for LLMs to be able to embed them in that way. We're also taking genomic measurements from the Parkinson's Progression Markers Initiative. And the main difficulty with these kind of, I guess, genomic features that we're working with is they're generated from whole blood peripheral samples. Being a neurodegenerative disease, we're using blood biomarkers, often taken from the arm, and then also, yes, we have cerebrospinal fluid data as well from that. So our hypothesis here is that there will be complementary information in these multiple omic markers, but what are the features of this which best guide the actual downstream classification task? So stepping back out, how do we generate these? How do we generate our network, our patient similarity network? So as mentioned, we have our clinical assessment, and we've done kind of several different iterations of this, looking at the different sections or the parts of the clinical assessment in Parkinson's disease. And we've also tested this on different LLM models, so kind of Llama, Mistral. And from each LLM, we generate a set of embeddings, and then we just measure the cosine similarity distance between patients. And this is simply just as our node. As in our network, we have 2 classes. So this is going to be a node classification task for our graph neural network. We are looking at Parkinson's disease patients and healthy controls. And the hypothesis here is really simple, is that when we use an LLM to generate these embeddings, we're going to get, I guess, distinct clusters. So you can imagine this single long line group of people here will have the same set of clinical symptoms for Parkinson's disease. And then you see that there's separate clusters here. You would expect those to have a different set of symptoms. And then finally, in the yellow, we see that we get a really strong group of healthy controls. So to reiterate, this is a classification problem, but we're expecting really high accuracy here because those who are healthy controls will not have symptoms, so they would expect them to closely together. What we're interested in is how we can guide our feature identification using this network for patient similarity. And that steps then into this deep learning multi-omic framework, or multi-omic idea. So how can we actually open up the black box of a deep learning model? And there's this PNET paper that came out a few years ago. And essentially what this tries to do is it tries to enforce a hierarchical biological structure based on the reactome database. Now, although the reactome database is incomplete and there will always be interactions that we can't account for, this is, at the moment, the best way that we can do to try and model interactions between genes pathways, and ultimately biological processes. And what essentially this is doing is encoding high-dimensional features, which we have to reduce down because of these genomic data sets. They can have huge, large number of features, and then we can represent not only the interaction, or not only the features which are important in each omic modality individually, but also we're hoping to capture informative interactions between our different omic modalities at the same time. And I'll talk about that in the next step as well. But really the important thing here is that we are doing a kind of a compression, a dimensionality reduction using a deep learning framework, which is kind of mimicking known biological interactions from the Reactome database. At the same time, on top of that, there's, I guess, no point doing any interpretation if you can't be confident in your results. So what we've done here is we've kind of done an extension of the gene set enrichment algorithm, whereby when we feed our model, we want to feed it 2 sets of genes. We feed it 1 set of genes, which we know are going to be related to Parkinson's disease, and then we kind of also give it a set of random genes. And these are just genes that have no historical relation to Parkinson's disease from anything that we could find in the literature. And then as a classic gene set enrichment algorithm, we ask our model, when you are interpreting which features are the most informative, we're hoping that it's going to identify the Parkinson's disease related gene set as those that are more important. And in this way, we know that once our model has actually performed a prediction and we're looking at the features it's identified, we have a bit of confidence that yes, this model is actually identifying and predicting genes which we know are related to our disease outcome of interest, in this case, Parkinson's disease. So finally, when we want to rephrase this as a prediction task, and in this task, we're using a graph neural network, but there is one slight kind of variation on that. And in this case, it is actually training the graph neural network in a kind of hierarchical training structure. So what that means is we're actually performing backpropagation through the network. So that's guiding what features we want to try and identify as informative, but also through our deep learning architecture at the same time. So we're training not only on the node features, but we're also training the deep learning encoder architecture at the same time in a hierarchical structure. So that means that they have a shared loss functions. It means that you're learning the same weights at the same time that you're training your neural network, you're training your deep learning architecture at the same time. So overall, in a very quick summary so far, we are predicting Parkinson's disease patients from healthy controls. We're trying to capture the similarity between patients, and we're trying to look at what are the features which are driving these predictions. And that's what we're most interested in. So kind of a couple of key questions and a couple of results that we're going to share with you is, so which LLM performed the best? And really, as you can see, all LLMs performed equally well. And the intuition behind this is that it wasn't really a difficult embedding task. In this case, it was really simple. The symptoms for people who have Parkinson's disease are really clear for the people who don't. What we're interested in is the actual added context that the LLMs can understand in saying that this symptom is actually similar to this symptom, and these patients are closer than patients who might have a different set of Parkinson's disease symptoms. Does the LLM outperform simpler approaches? Yes, barely. If you just do a logistic regression to try and see which are the key features that are going to separate a Parkinson's disease patient from a healthy control, you do get pretty high similar accuracy. But in the next step, what I'll motivate is that actually the LLMs have an added statistical significance in their interpretability. Another interesting note that we found is that which actual sections of the Parkinson's disease, so in our clinical assessment, as I mentioned earlier, it's been split into 4 parts. We looked at parts 2 and part 3. Part 2 cover non-motor symptoms, part 3 cover motor symptoms. What we found was that actually part 3 seemed to outperform even just the combination of part 2 and part 3 together. And what that kind of intuitively says is that actually the key symptoms which we're looking at in Parkinson's disease are the motor symptoms, and that the non-motor symptoms seem to maybe be also kind of this, I guess, a complementary effect or kind of an added effect to the actual key motor symptoms of the disease. Finally, when we want to actually look at the interpretability, and this is the kind of, I guess, the main important takeaway, is that we were only able to actually achieve statistical significance in that kind of comparison between the features of the Parkinson's disease related versus the non-Parkinson's disease gene set. So only when we were actually able to combine, only when we used the LLM embedding models were we able to get the context that was able to actually accurately identify groups of Parkinson's disease patients who had phenotypes which were able to achieve significance. And also what we found was that early in disease, so we actually did this longitudinally, we looked at a few different time points. We were only able to find this at about year 2. So we needed to wait for a bit of a stronger disease signal in the kind of omic or genomic data sets before we were able to actually reach significance. And then, yeah, we found a couple of genes which we were able to find also in literature and also some pathways and processes. But really the key take-home message here was that only using the LLM embeddings were we able to actually get the kind of contextually aware significant results. So again, this is the final overview, and just the paper is currently on medRxiv. It's currently going review. Untangling heterogeneity in Parkinson's disease is really, really hard. This is a nice way to kind of, I guess, group together phenotypes or patients with similar phenotypes. And thanks very much. Be happy to connect afterwards.
Thank you. Thank you, Ryan. Again, I'll ask people to connect with you afterwards. We're out of time again.